This is the sixth part of our Enterobacteraceae lecture. And in this lecture we are going to talk about several organisms. We're going to start out with the members of the Klebsiella family, or I should say tribe. It's the Enterobacteraceae family, and then several of the organisms are within tribes. So the genera that are in the Klebsiella tribe are the genus Klebsiella, the genus Enterobacter, the genus Pantoia, and the genus Hafnia. We'll start out by talking about Klebsiella. There are numerous species of Klebsiella, including Klebsiella pneumoniae, Oxytoca, Klebsiella pneumoniae subspecies Ornitholytica, Klebsiella planticola, and Klebsiella terrigina. Now, Klebsiella is non-modal. Put a star around that. It is one of the organisms that are in the Enterobacteraceae that are non-modal. Shigella is also non-modal. Yersinia pestis is non-modal. Most of our other Enterobacteraceae members are modal. Klebsiella pneumoniae is the most commonly isolated species of Klebsiella. It has a polysaccharide capsule, so it's an encapsulated organism, and because it's a capsule producer, it generates a very mucoid colony on agar media. You will see a lot of Klebsiella in the clinical lab, and it really is a mucoid kind of no other very viscous organism on agar media. It is a frequent cause of lower respiratory tract infections in hospitalized patients as well as immunocompromised individuals. It can cause many other types of infections. It can cause wound infections, of course bacteremia, and it also causes urinary tract infections. It has antibiotic resistance due to plasmids, and it can transfer these antibiotic resistance genes to other organisms via the plasmid, which can lead to drug-resistant outbreaks. So here's Klebsiella, and like I said, it is a capsule-producing organism, and on all different types of media, the organism tends to be very mucoid looking, very mucousy. So in the right upper corner, you could see it on blood agar, and in the center left, it is on MacConkey agar. It is a lactose fermenter, so you could see on MacConkey agar, it's bright pink. Here is a capsule stain, and you can see if you use a capsule stain, the organism will stain and the background will stain, but the capsule will not. So you'll see a nice white clear halo around the organism. We're now going to talk about some other organisms, which are Enterobacter, Cronobacter, and Pantoia. So we have the species Enterobacter cloacae, Enterobacter aerogenes, Enterobacter gergovii, and what used to be Enterobacter sakazakiae. However, it was renamed to Cronobacter sakazakiae. So Cronobacter is a newer genus. This organism has a yellow pigment. There's Enterobacter Hermacei, and what used to be Enterobacter agglomerans, which has been renamed to Pantoia agglomerans, which also has a yellow pigment. The general features of these organisms are they are modal. So Klebsiella is not modal. These organisms are modal, but they do have a morphology similar to Klebsiella on MacConkey agar. Enterobacter cloacae and Enterobacter aerogenes are the most common organisms that are associated with human infection, and they've been isolated from wounds, from urine, blood, and cerebral spinal fluid. And here you have a mucoid yellow pigmented colony of Cronobacter Sakazakiae, and this is BHI, or brain heart infusion agar, which is a non-selective agar. Here is the yellow pigmented Pantoia agglomerans, and this is on sheep blood agar. We're now going to talk about the genus Serratia. There are numerous Serratia species such as marcescens, liquefactions, rubidii, odorifera, ficaria, fonticola. In general, these are opportunistic organisms. All of the species except for Serratia fonticola ferment lactose slowly. So they are slow lactose fermenters and are ONPG positive. They also produce DNase, which is an enzyme that can break down DNA. They are highly resistant to antimicrobial agents. Some of these species have a pink to a red pigment, so they can be very bright red, especially if they're incubated at room temperature. So if you take Serratia and you put it on a general nutrient agar and incubate it on a bench top, some of these species will turn very, very bright red. Serratia marcescens is the most common species isolated, and Serratia has been isolated from urinary tract infection, respiratory infection, bacteremia, as well as septic arthritis. And here you could see the Serratia marcescens growing on a blood agar plate on the left, and that blood agar plate looks very hemolyzed. In this picture it looks like chocolate agar. And on the right it's on McConkey agar, and you could see the bright red pigment. We're now going to move on to the last part of our Enterobacteraceae lecture.